Welcome to Nikolai's genetics lessons, and today we are going to talk about bioinformatics and conceptual idea of alignment. Alignment can be global and local. So, alignment is a task of locating equivalent regions of two or more sequences to maximize their similarity. Compared to sentences, computational biology. So, one and second. In red here we show mismatch, where, for example, if we compare two sequences of proteins, it can be two sequences of DNA strands or two sequences of RNA strands. So when we compare these two sequences, we may have mismatch. And sometimes we may also see that the whole fragment is missing. So we call this gaps. So mismatches and gaps give us idea how two sequences are similar to each other. And then we can compare, for example, the first sequence with another one, not this one, but another one. For example, between 2, 3, 4, 5, 10 different species or varieties of, same species. And we can organize them according to their similarity. For example, we can build a phylogenetic tree based on this information. Alignments of related sequences is expected to give good scores compared with alignments of randomly chosen sequences. For example, if we compare sequence of dogs and foxes who are related, we are going to get much higher score than if we compare sequences of dogs and elephants. And in practice, the correct alignment doesn't necessarily have the best score, since no perfect scoring scheme has been devised. But using general rule, we can say if two sequences are more than 25% identical, they are likely related. If two sequences are between 15 and 25% identical, they may be related, but more tests are needed. And if two sequences less than 50% identical, they are probably not related at all. Now let's talk about two types of alignment. And alignment can be global and local. So this is example of the global alignment, and this is example of the local alignment. The goal of global alignment to align the entire length of two sequences, maximizing similarity across the entire length. And this is suitable for comparing closely related sequences of similar length. We're using this for identifying overall similarity and evolutionary relationships. And example would be aligning two full-length protein sequences to assess their overall evolutionary divergence. When we compare sequences using local alignment, we're using different goal. Goal would be to identify regions of high similarity within the sequences regardless of their position within the overall sequence. And this is suitable for finding short conserved regions or motifs within a longer sequences. Here is an example. Take a look at this protein, and if we analyze this protein in different species, we probably will find that in many places there can be mutations found here, but here is an active site, and most likely this active site is going to be conserved. So we will see picture that in this site we are going to see the same sequence across all the species. If we will analyze the whole sequence of this protein, we'll probably find some mismatches if we compare different species, some gaps we also may find here, but if we'll find that in many species some sequence is conserved, this probably means that this region is very important, and probably this is active site in different proteins, related proteins, enzymes in different species. We're also using local sequence analysis in order to find distant evolutionary relationship where only small regions might be conserved. And my last comment would be that for global sequence analysis, we're using Needleman-Wunsch algorithm, and for local sequence analysis, we're using Smith-Waterman algorithm. And in my following videos, I'm going to talk about pairwise alignment and multiple sequence alignment. And this is all for today. See you in the next video. Goodbye.